PhD and postdoc in there are initially some PhD and postdoc in the urban lab. They actually find out you can just use sting agonists, put into tissue media, or you inject into the animal. They're able to actually sting, which is a mystery. So I started from there, like trying to know how supposed to be intracellular ligands able to active sting when you just put them in the media or in the in the animal body in the circulation system. And then initially I did a screening trying to find a CGAM transporter, but unfortunately that got scooped when I was at my year 3 or year 4 of my PhD. So and we actually got a transporter, but one Stanford group, Lin Li, and one UC Berkeley group, David Rollet, they did a very similar assay, and then they published very well. But then at that time, I was like, okay, now I need to find a new direction. But at that time, I already read a lot about the STING pathway, and I read a lot about innate immune pathway, and I found like how STING is able to induce this. They're like the general theory about STING seems kind of clear, but then it's just a mystery about how STING can do such a diverse output. And then aided by the new cryo-EM structure of STING. So we're like, oh, what would be the simple theory can explain all the phenomena? And that's having the seed in my mind. And then we have the theory around like 2020, early 2021. This is the channel, but it takes a lot of effort to realize there's a perfect perturbation for us to quickly confirm that's the right direction. So we started doing the actual experiment at mid of the 2022, which is roughly mid-September. And then it's actually unique. Nearest is very unique in the biological paper because from the first experiment to our first submission take 2.5 months, because all the experimental result matched to our theoretical prediction very well. And then it took us some effort to build up the liposome experiment, because that's the part we have no experience. But then we, again, a PhD student from Urban Lab, Ivan, who is super smart, and then he helped a lot on the liposome experiment. So that made us actually be able to finish a proto-liposome experiment in 3 months, although none of us have ever done channel proto-liposome assay. So it's really a teamwork. I happen to find the right people with skill set to help join force to prove these things together.
Yeah, that is amazing. As you mentioned, that as you start working on the property of STING, as I put in the intro of your seminar, this is the extensively studied protein, and people have solved its structure and have been studying its function. Does it come as a surprise to you that it might be a proton channel? When do you have that idea? And do you have any self-doubt in the very beginning? And could it be true? Because my PhD advisor, like he's the most humble scientist. He'll say, if you make something like so out of the box at this caliber, we'll say, first ask yourself, how could no one else have come to this conclusion? Is it because this question itself is not interesting, or is it because other people don't have the tool to do this, or whatever? Like, have you any doubt, or what is the thinking back then, and how did you tackle it?
Yeah, I think that's really quite a good question. That's if anything, we keep doubting ourselves. So first, is it even wise to choose STING as a research field? Because when I started, when I do the transporter of the STING agonist CGAM, that's actually not bad, because not too many people focus on that. But of course, later I learned many people focus on that. But then to actually focus on STING, it may not be the best thing, because especially for a lab, we don't have too much experience in the STING field. So it's can be hard, especially compete with people who are already in the field for like 10 to 15 years. And then the second question is like how we actually come to the hypothesis in the channel. So when I actually later share the story, it will look different. It look more of a making sense step by step. But the actual thing is when we read papers about in a lot of random papers in innate immunity, we start to realize that all the supposedly diverse downstream stimuli can also all be explained by a single upstream like cue, and then the single upstream cue can be explained by if STING in the channel. And so that have several jump a lot of different hypotheses. But when we figure out if seeing the channel, it just explains everything perfectly. And then we're like, okay, this is really beautiful, but it's a little bit too far away from the known evidence. And thin field, there are always contradicting evidence. So I rely a lot on the genetic evidence, which is is very easy to replicate, and then it's also making more sense to interpret it in a systematic way. And some paper even not published very well, like from the journal wise, right? But then they actually really solid, good papers. And I will actually mention, I think at the last slide, for example, but also I think when our paper got published, this is almost at the time when the field had enough evidence, so people start to realize the channel. Because for example, after our paper got published, there's immediately a bioarchive claiming similar conclusion as us. And the funny part is when I emailed Richard Euler, which is a professor who published a really like great GCD papers 2020, Fisher 2020, the first author. It's like I basically wrote an email, thank him and his group for contributing to the theoretical foundation. And Richard, I literally can share the email, but basically Richard, their group have a similar hypothesis. They actually almost want to try, but then they didn't really try. But so I was thinking the evidence accumulate to a level that I think people all start to think about. Maybe it's too messy. Maybe there's a very really simple way to explain everything. And then we just maybe have the theory a little bit earlier than the general field. Yeah.
And just to briefly comment on what you have mentioned, basically I will attribute the discovery to like your courage as of the group or your collaborators that outsider of the field to challenge the paradigm, and also at the perfect combination of the expertise of the people in the team, and also the tools, as you mentioned, there's a chemical tool you're going to mention a little bit in your presentation. So these all perfect combination that lead to this discovery. So without further ado, I will give the stage to you, and afterwards we can have a discussion and invite all the participants to do question and answers. And if anyone has question along the way, I encourage you, instead of cutting into the conversation, because like we are doing Zoom, it's not clear who is going to speak. So to maintain this order, maybe you can type in your question in the chat box and we will get to you when we finish. Is that okay? Or you can also raise your hand at the end when we are done with Bingxu's talk. Okay, so I will give it to Bingxu.
Perfect. Thank you again for giving the chance. So I hope to keep it short and we can discuss more. Bingxu again here, and so I got trained at MIT biology, my PhD near Koch and their Irvine group, and now just starting my postdoc in David Baker's group. To start with, I think, as I mentioned to Charles, evolution is the most exciting, evolution sensing is the most exciting problem for me. And I think, in all the different biological system, host pathway armories is one of the most exciting like branches in the biological systems. Yeah. This armories really yield diverse immune system trajectories. And this is such a basic thing all the biological system need in bacteria. Even like as simple as bacteria, we need to sense and defend against the invading pathogens, which is phage. And in human, complicated organism like human, the sensing and the defending will be even more complicated, where we're supposed to defend the viruses, intracellular bacteria, fungi, and the parasites of different sizes and different patterns. And then the interesting part is this, a very extensive armories really give us a lot of different trajectories, like, coming from bacteria to human, where this immune system come from, and what's a new thing we aid to achieve a broader recognition and defense during these processes. So that's really will give us a lot more information about general principles of evolution. And then Sting, after we read enough about it, Sting became a very obvious target for us to research on, because they actually have the both properties. So Sting pathway show both like extreme conservations and the extreme innovations. The conservation, I hope I can convince you, it's very obviously from this side when we compare the bacteria STING structure to human STING structures, the conservation site is the ligand binding domain here. That's shown here is a ligand, and then you don't even need to be a structure biologist to see the good similarity between the bacteria ligand binding domain and a human ligand binding domain. But when we are talking about the innovation part, so bacteria linked to this ligand binding domain, most of bacteria have a TR domain, Well, in human or like the relative in human and other animals, like we actually gain this transmembrane domain. And at the time, before our work, we still don't understand, why the function of this transmembrane domain. And then despite the similarity of the structure of the ligand binding domain, the downstream downstream functions are also quite different. For bacteria, primarily when the STING got activated, you have the cell death. But for human, it's really diverse. You have interferon, you have NF-kappa B activation, you have non-canonical autophagy, and you have inflammasome activation. So it's really interesting how supposed similar like signaling platform can be really still around and then put in new functions, put the new functions on them. And also, it's not only a very evolution interesting, question for us researchers on. STING signal also really important for the physiology of homeostasis. Supposed to be one important protein in the double-strand DNA sensing. It's really responsible generally for our body to sensing specific pathogens. And also during, when we have a genomic damage or mitochondrial damage, there are some free DNA leaked out from our genome, and then that's really can trigger the STING activation. And for the downstream effects, it's really important for activating STING, play a really important role in host defense, defending against invading pathogens. Also have been shown to have really important function in anti-tumor immunity, and its dysfunction also can lead to autoinflammatory diseases. So that's like combination make us a perfect way for us to go into and research on, how STING can achieve such a goal. And when we started to research on this problem, the field have a general good understanding of how STING, the general, working philosophy of the STING pathway. Basically, when we have a bacterial invasion or when there's a double-strand DNA exists in the cells, they both can lead to the existence or appearance of a cyclic dinucleotide in our cells. And the cyclic dinucleotide can serve as agonists to bind on STING. So normally when STING is in apo state, resting state, is a membrane protein on the ER membrane. Upon binding to the agonist cyclic dinucleotide, STING will start, go through a conformational change and then start to translocate to Golgi, where it start to oligomerize and then give the downstream functions, including interferon and the non-canonical autophagy and NLRP3 inflammasome activation. Well, people have generally a pretty good understanding of, the how STING induce interferon. It's having a long mystery that how STING can induce non-canonical autophagy and NLRP3 inflammasome activations. So, and it's even more, interesting of like how a simple small protein can actually give such a diverse responses, right? But as I mentioned to Charles, it's not that diverse to to think about. It's not completely different downstream pathways. And when we are talking about the unknown, the functions that we don't know how STING activates them, specifically non-canonical autophagy and inflammasome activation, they're actually triggered by the same environmental signal. It can be triggered by the same environmental signals. So the idea is when we have acidic Golgi or endosome, and then it have a high proton concentration inside at the low pH, and when we have a pathogen invasion or just a simple chemical or physical damage to damage these organelles, we'll see a pH increase inside organelles, and then its pH increase can be sensed by downstream effectors to induce non-canonical autophagy and NLRP3 inflammasome activation. And I want to highlight the two papers really inspire us, because this although the downstream effect, the non-canonical autophagy and NLRP3 inflammasome activation have been known for a very long time, but the these two papers really tell very clearly how it actually happened from a pH increase, a pH increase to the two downstream effect. As I mentioned earlier, that we know STING can activate the both downstream effects, right? So the obvious question for us is, does STING actually induce pH increase, the proton leakage, to further activate the two downstream pathways? And if that's our hypothesis, what's the best way to answer that? And because we are thinking about STING connecting the middle step, we are hypothesizing the middle step between these two steps are the pH increase. We really need the organelle pH reporter to answer this question. And the reporter we decided to use is this genetically encoded pH sensor. And the basic structure is it fused two fluorescence protein. One is pH sensitive GIP SAP, and one is the pH insensitive, the ref fluorescence protein MRuby3. So upon acidic pH, we will see a quenching of the green fluorescence protein, while the MRuby is still kept the same. And when the environment became the neutral pH, we see the SAP gain fluorescence, and then MRuby still as internal control, internal reference. So basically, the change of the fluorescence between SAP to MRuby can serve as a good reporter for reporting the pH inside a given organelle. And imagine the good part of the genetically encoded pH reporter is you can really fuse with different localization sequence to put into a different organelle. For a given organelle, if it's acidic, we will see a quenching. But if there's suddenly a pH change, we will see the increase of the green fluorescence protein. So now we have pretty good reporters. It's time to answer our question. Does STING activation actually increase the pH inside a Golgi? So here is the simple experiment we did that basically we construct a cell line, a STING expressing fibroblast cell lines, and then we put this genetically encoded pH reporter into the Golgi lumen, and then basically we are measuring the SAP versus MRuby3 fluorescence protein. And the more neutral the pH is inside the lumen, the stronger of the yellow signal. And then we have two groups. One group we just put DMSO, the other group we put a STING agonist diABZI. And then we're asking a simple question is, when we active STING, will we see a pH increase inside the organelle? And I hope it's pretty obvious for you that when we put DMSO, we didn't see much change, but then upon STING activation, there's a very bright signal increase, just indicating there's a pH increase inside a Golgi lumen. But then the next question is STING actually present the vesicles to have the pH change, or is STING doing something different and to induce this effect indirectly? And you can imagine there are two downstream theory here. One is when we give STING agonist, STING translocate to the acidic organelle, and that's somehow induce the proton leakage, or it could be STING translocate to some organelle and then give some secondary metabolites that indirectly induce a different acidic organelle proton leakage. So how would we distinguish that? We decided to take the super resolution microscope and then to image the time series upon STING activation and pH change. And it's pretty obvious for us to see that for all the acidic vesicles that have pH change, means they have a light up SAP signal, they always have STING on these vesicles. And for the vesicles that don't have SAP, that you can see they have MRuby but don't have SAP, so they don't have STING. So STINGs strongly on the vesicles have a strong localization association with the vesicles that have pH changes. So that support more of the first theory, that it's highly likely that STING translocate to a given acidic organelle and induce a pH change in that specific organelle. And next question is, it's like we know it's as a fact, as a phenomenon, but how does STING actually induce a pH change upon translocation? As I mentioned to Charles earlier, I think it's not very obvious to hypothesize STING is the proton channel. A more likely situation is upon STING agonist stimulation, STING translocate to a given organelle, and then probably there's known proton channel there that somehow STING is able to activate it, and then they induce proton leakage and to induce downstream pathway. So when we have this good hypothesis, we just want to hunt for a gene. The most obvious thing for us to do should be the CRISPR screening, right? And for doing the good CRISPR screening to find out the unknown protein channel, we really need a good reporter system to do this. Proton leakage is not a really good reporter for genetic screening, but proton leakage can induce downstream non-canonical autophagy, and we have a lot of good reporters for non-canonical autophagy marked by alcyl-3 lipidation. So our general, we decided to use the downstream pathway to try reporters to try to find out the proton channel. Imagine upon STING activation, when we knock out this proton, unknown proton channel, it should not be able to induce a non-canonical autophagy, and that's how we can tell if we got the right proton channel or not. So the simple experimental workflow is we have the cells that's expressing STING, and we transduce the cells with the pooled sgRNA library, and then we get cells that with diverse knockout, and then we give STING agonists to stimulate the cells, and then we permanent cells and to sort the cells with high LC3B signal and low LC3B signal. Imagine if you are the cells, if the cells have the right knockout of its proton channel, even upon STING activation, they should not able to induce the downstream LC3 lipidation. So they will be more enriched in this population versus this population. So when we did the experiment, we're trying to sort out, we are trying to find out what are the cells or what are the genes, like what are the gene knockout that make cells more distributed in this population. And the screening is beautifully done, and we almost uncover all our positive controls. Basically, the green genes here are the all the ATP, all the ATG, functional pathway, and the cyan here is ATPase. So these two complexes are known to sense the proton leakage and to induce the downstream non-canonical autophagy. But even if the screening is so well done, we uncover all the positive controls, we still don't know the initial trigger. Like we didn't find any, known proton channels like in this arm that can explain the phenomenon. So this is a pretty awkward situation. We did a beautiful screening, but we didn't find the thing we want to find. And I want to quote one of the collaborators who support us during the process, Professor Paul Blaney. And Paul says the maturing in the functional genomic field, while the pool screen data are sufficient and reliably to set out the direction of focusing investigation, even for negative results. So here is like pretty conclusive negative results, and we didn't find the proton channel, but we know we did such a good job, and it could be a possibility that there's just not a unknown proton channel mediating this process. And alternatively, it could be pretty crazy, but it could be just STING translocation and somehow be able to mediate the proton flux. And that's the reason why we cannot find it. And well, this is an unknown thing in the mammalian world that we don't know if there's a channel or innate immune sensor can serve as a channel, but it's not something like unheard of in the general biological world. At that time, I read a lot of papers about the plant innate immunity also, and in plants, people actually find out several really interesting innate immune proteins can, upon sensing a danger signal, they can serve as a channel. So I was like, okay, plant can do it. Maybe the mammalian cell can do something similar, and that's just give us a little bit confidence that is a possibility. Like at least biological system can use channel as innate immune system. We had no evidence for the theory, but then when we realized, oh, we actually have the full STING structure, and then how about we just do a quick analysis to
Are you muted? Sorry. I will ask him. Sorry, I will ask him. I'm checking in with him. I think he just get disconnected for a little bit. Sorry for this. Anyone has question? So he's trying to reconnect. They have some Wi-Fi issue, so let's just be patient for a minute. I think he will be with us in a bit. Is he back on? Yeah, if you have questions, please feel free to type in now. I will just also take the opportunity to give a bit of a introduction to our journal club, and we are still trying to explore, like, specify the time and the agenda, but generally we want to take this opportunity of the journal club to discuss some of the classical paper in the field of modern molecular biology, neuroscience, developmental biology, especially, or maybe also include sometimes immunology. So this will all be okay. Let me see. Hi, hello.
I think the UW Wi-Fi is stuck.
Okay. Make you co-host.
Yeah.
So we all said now you can share your screen. Yeah.
Yeah. Let me try to see if I can use my phone hotspot.
Okay. Great.
Okay. So I'm glad. Thanks to 5G. So that's actually give us a cool first hint that there's actually a central cavity, at least inside the sting, right? So we got pretty excited because we know nothing about this ion channel. So, but what we know is this field is very technical. We need to ask the right expert. So when we consult Professor Chris Miller, you have this very philosophical, quote. It's like positive result here cannot guarantee the channel, but negative result also cannot exclude it. So while the in silico prediction really give us some evidence, we really need to find a good way, to test our hypothesis. And simple thing is, can we actually really find a good way to block the proposed pore to test our hypothesis here, right? And then we really have this theory maybe around early 2021, but we just can't move at all, for the whole year because we don't know a really good perturbation until like maybe I think it's around April 2022. And then this paper published that surprisingly find a new sting agonist binding sites. So this is the drug from the like Indian company that they trying to screen for agonists for sting to produce interferon. And then they actually got a really weird, interesting compounds. And then the structure are solved by the UT Southwestern groups that they find instead the traditional binding spot like cGAMP or the diabezia we use in the ligand binding domain, right? Called ligand binding domain. We actually, the 53, the new small molecule, the company screen, they actually bind the transmembrane domain there. And then this is quite a new binding pocket people have never known before. And in their paper, they're like, oh, we don't know. Maybe this represented a new, agonist binding site. And for us, we got really excited because this is exactly the central cavity bind to is exactly the proposed, the pore area we were thinking about. So this could be a perfect perturbation to block the pore function sting without blocking other arms like interferon induction. Because 53 is already proven by their data that it is it is a agonist for STING interferon production. And then we actually negotiate with the company who have this compound for 3 months, but then somehow because of MTA issue, they actually the deal was broken, and got really frustrated. And then I was like trying to go online to search where other sources I can get C53, and I realized the chemical just started to sell it. So using $50, I bought a lot of C53, and that can make me start to do experiments. A simple question to follow is, can C53 really block the poor function of STING, right? And then I have shown you this assay before. Basically, we have a STING agonist stimulation, and now we have the STING like suppose a channel blocker C53. Can the C53 combination with diabetes really block the STING agonist alone? And it's pretty clear when we use the drug combination or like C53 on top of STING agonist, while the STING agonist diabetes alone is able to induce very strong pH increase, but when we use C53 combination with diabetes, it's almost completely blocks the pH increase inside of acidic Golgi, and that's really give us a strong confidence that it's likely C53 is blocking the role the function of C53 is able to bind this proposed poor areas, able to block the pH increase in this system, right? And but the next question is, the C53 is a new molecule. We really don't know if the STING transmembrane protein, transmembrane site is only pocket, or probably it could be C53 bind to an independent protein and somehow is able to change the pH increase inside a acidic organelle. What we do is, from the paper before, they already made a mutant of STING that still can be activated by the cGAMP or the ABZI, but then they mutate the binding site for C53, so C53 is not able to bind them bind the mutant STING very tightly. And so for wild type STING, we already know that you give normal STING agonist able to have a proton flux, and then when you give a combination of STING agonist plus C53, you don't have this proton leakage anymore, or have a really reduced proton leakage. What will happen for the mutants as mutant STING? Supposedly, if C53's function is really through binding to STING, we should see for the mutant STING, when we give STING agonist, we're able to see the proton flux. But when we give the C53 plus the STING agonist, because now C53 cannot bind to the mutant STING very tightly, we will still see the proton influx here. So did the experiment actually match our prediction? Yes. So for the wild type STING, as I showed you, there's a pH increase indicated by the SAP versus mRuby signal. We see a pretty good increase, and the increase is blocked by co-treatment with C53. But when we have the STING mutants, the mutants that cannot bind to the C53 very tightly, when we give a diabetes only, we see a pH increase. But now, when we add the C53 on top of it, we're no longer able to see the blockade effect induced by the STING agonist. That's really give us a lot of confidence that C53 blocks the pH increase by tightly binding to the transmembrane domain of the STING. So this is all good, but this is all cellular data. So there's 1,000 different factors can influencing it, and the ultimate role in the ultimate assay to prove if one thing is ion channel or not is able to purify them, and to see if the protein itself is sufficient to give us ion influx in this process. So the central idea is able to make a protoliposome assay. So the general simplest schematic is we have liposome and we have purified STING, and then we load the purified STING on the protoliposome, and we're trying to measure if the STING can meet the proton leakage in the process. And the more complicated the actual process is, when we have the protoliposome, STING containing protoliposome, the liposome is already preloaded with high potassium buffer inside. And then we also have the pH sensitive dye ACMA that at a neutral pH is able to freely travel in and out of the liposome. And when we add valinomycin, which is the potassium ionophore, it can mediate the driven by the concentration difference of inside or outside the liposome. The potassium will leak out from the liposome, and then the leak out process build up a voltage difference between the intro and between outside of the liposome and inside of liposome. And if STING indeed can mediate the proton, can mediate proton translocation, we can imagine driven by this voltage difference, the proton will rush into the liposome to acidify the liposome, and the ACMA dye can be sequestered and quenched inside the acidic organelle. So we will see our fluorescence quenching in this environmental system. And at the end of the assay, we add CCCP, which is a proton ionophore, as a positive control to show that our system really work well during the whole assay process. So the prediction is, normally we have a fluorescence liposome. If the staining containing proton liposome really can mediate the proton translocation, we will see a quenching of the fluorescence here. And if CC3 really can block the stain proton translocation effect, we will see a reduced quenching when we treat this thing proteoliposome with CC3 molecule. And the result indeed match our predictions. So when we give vancomycin, for the just empty liposome, we see no big change of the fluorescence. But then when we have the stain containing protoliposome, we see a pretty good quenching effect when we give vancomycin. So that really tells us that STING can mediate this translocation by proton translocation. And more importantly, when we co-treat the STING with CP3 molecule, we see the quenching effect reduced. So that's really proving our predictions right, that STING can, on the protoliposome, can mediate the proton translocation, and then co-treatment with CP3 really can, reduce its translocation effects. And we have been in molecular details a lot that are trying to see if STING can induce the proton change. But initial our motivation is try to understand how STING activation is able to give two downstream pathways, right? Now, since we have CC3, it's really good perturbation for the Golgi pH, the proton leakage. The obvious question is blocking the Golgi pH change can really blocking the two downstream functions. So the first assay is, we know the non-canonical autophagy basically can be marked by the LC3 lipidation, means the free LC3 now can be decorated or lipidated to put on the specific organelles. And then, as we can see here, when we have the reporter cell line with STING HA and LC3 RIP, for the untreated cells, we didn't see much of the LC3 decorated on the vesicles, but when we give the STING agonist, we see a pretty strong LC3 lipidation on this specific organelle. But then when we co-treat with CP3, we still see the STING translocation into the organelle, but we no longer see the LC3 lipidation on these specific organelles. That's really telling us, yes, CP3 is not only blocking the pH increase in these organelles, but also blocking the downstream non-canonical autophagy pathway. Similarly, for the NLRP3 inflammasome activation, is also upon the activation, or during the activation process, NLRP3 translocate from cytosol onto the specific PI4P enriched Golgi vesicles. And we can see when we treat cells with diabetes, we can see the NLRP3 amnion green can enrich on this STING containing vesicles a lot. And then when we co-treat with CP3, we no longer see the enrichment of the NLRP3 on this STING containing vesicles. That's really tell us that it's really consistent with our theory that indeed STING is inducing the proton leakage to increase the pH and to give a downstream effect, and specifically blocking the pH increase effect of STING can specifically block two downstream pathways that previously people don't know how this is being induced. And now, starting from the simple mystery inside the field, that's how STING is able to induce a two downstream pathway. Now we show that it's very simple that upon activation, STING able to translocate on these acidic vesicles, induce a proton leak, and to induce the two downstream pathways. So that's like really complete the picture of the STING field to explain what's going on there. So I want to thank for all the members who are directly involved in this project. My advisor Nir and Daryl will give a lot of support, and my co-first author Rebecca Carlson. Rebecca basically did all the imaging work, including the live cell imaging and later other like images. And then Ivan is responsible for basically we did side by side for 3 months or even more to trying to build up the initial the protein liposome assay. And Matteo, who is expert, we work on STING together from the day one probably, and then and later Matteo really helped a lot for the inflammasome pathway assays, which I actually didn't show too much, and all other Ali, my undergrad mentor, and then Katan, Mark, and Matt involved in this process. And for external collaborators, Chris Miller have really be our external advisors to teach us so much about the channel field, and Paul Blaney for supporting us for the imaging. Professor Chonggang Gang from Tsinghua University and Wentao Huang have been basically teaching me a lot about protein purification. I never purified a protein before from mammalian cell, so without their help, we really cannot make it happen. Thank you.
This is terrific. I see we start to have some questions. Before getting into that, I just hope you can clarify some of your last slide. Like you say this inhibitor blocking the channel function of STING. Does affect the inflammasome, does affect the autophagy aspects of the downstream signaling. What about the interferon? Does it also affect that, or is spells on part of the function, downstream function?
No, the interferon, if anything, the interferon function is more of like, interferon function is actually kind of independent of this process. People understand very well. Basically, STING have a C-terminal tail. The C-terminal tail have a TBK1 binding site, and then basically when they oligomerize together, they can recruit TBK1 to do the cross phosphorylation. And when I say people understand very well, is people can even build up something to just link the C-terminal tail with some random with FKBP12 protein. When they oligomerize FKBP12 protein, they can actually induce interferon. So it's really nothing to do with
so you probably also can test in your system, kind of serve as a negative control, like you say, oh, it's very specific because normal the one aspect of function is unperturbed.
Exactly, because you can people can always criticize, oh, the CP3 probably can block everything or just make cell unhappy, right? But then every time so here's the thing, if anything, when you block the two other functions, you get a slightly increase of the phospho TBK1 or phospho STING, which is the early indicator of interferon induction. So that's actually a good thing. You're like, okay, it's not like STING activation is reduced, so you see the reduced downstream. At least some are even slightly enhanced.
Oh, we see Shanshan have questions. Shanshan, are you online? Are you there? Do you want to unmute yourself? Let me see.
Hi. Hi.
Hello.
Hi. Great talk, by the way. I'm just curious how because I'm also working on ER, and recently STING pop out in my experiment. I see some protein changing in my experiment, so I'm curious about STING protein turnover regulated on both ER and Golgi, and also, for example, when traffic to Golgi after the proton going out, will it be degraded or will be translocated back to ER for recycling? Thank you.
Yeah, so that's actually I said I collaborate with Matteo, or one of his papers is actually focusing on how STING being degraded, and there's also there are other papers in the field that really want to focus on how basically after STING activation, STING will traffic from ER out of ER to the Golgi vesicles to MVP endosome, and the degradation details is more complicated to explain here, but there are several literature now to basically argue after STING activation, they somehow be ubiquinized and then be degraded in that MVB. And then so that has been a working hypothesis, and we have some evidence, and other people also have different theories, so you can check that out. But that's the degradation of STING is pretty important for terminating the STING response. If you actually block the pathway, you see a dramatic more of the phospho-STING and also interferon production there.
Another question is about this liposome assay. Well, I've never done a liposome assay, so they say no nothing. But I'm just curious, like you use like put the potassium inside, and then let the potassium to go out to use the drug, and then you can, because of this gradient going down of potassium, and then you see the proton goes in. I'm just wondering, do you need to first like prime STING with the agonist to first activate STING before you put on the liposome, or it's just like a dead STING can do this? Second question is, can you do this? Let's you put STING on the liposome and you treat with the agonist to induce the proton. This is more like physiological, like what's happening.
Yeah. This is like a really good question. If anything, that's only phenomenon like trouble me so much during the revision process.
Yes.
That we really don't need agonists to induce this. Like normally for all the data I show, like all the experiment, all the phenomena, we really need active STING, and then to put them, be able to have this pH increase in cells, right? But on liposome we don't. So and it make me wonder if if the channel is just like driven by so when we give in cells, when we give agonist, is a function primarily it's like when they are activated they can translocate from ER to Golgi, which have a bigger pH increase inside outside the lumen, or we are just observing an artifact on liposome. Maybe the curvature of liposome somehow makes the STING open. We don't know. I'm slightly biased to like we really don't need the conformation change itself to make the pore open. We just need a voltage change, but I don't have solid evidence for that.
Experimentally, is just to add on this design of here, like comment, is it possible to add the agonist to your liposome system?
Yeah, we added and we actually show like it doesn't make any difference. So we tried the natural agonist CGAMP, we tried the synthetic, because natural CGAMP the penetration of the liposome not so good. So instead we also give a diABZI, which is they can freely penetrate the liposome, no difference. And I think the other group basically later there's a bioarchive, they basically also did liposome experiment. They also didn't really see a difference of aiding agonists. So it's really a question: the STING itself function as a channel, and basically the environmental cue to decide the channel is open or not may not have anything to do with the agonist binding induced conformation change. May just because the agonist binding induced conformation change, but the conformation is just supposed to bring STING to the Golgi. So the channel function itself, whenever you have enough gradient or voltage, they should be able to do the job. Or the other paper they explain in a way that maybe just experimental artifact. Maybe during the purification somehow things activated, or the curvature, other things, the lipid composition. But this is really a mystery when we do the experiment. Thank you for bringing that up.
Thank you. Oh, just one follow up.
Yeah.
When you STING is activated, I remember it goes to dimerization, right?
Oligomerization. Yeah.
Oligomerization.
Yes, STING itself is dimer. Yeah.
So are you saying like this oligomerization is not required for the proton to go through? Okay.
We think so. We think so because on the liposome we didn't really give any agonists, and then I think personally oligomerization is more important for bringing them to the acidic organelle. That's like trigger for the translocation to make them translocate to the organelle. But again, I think people always have this argument is without agonists can STING oligomerize. But I personally I think for being function as a channel you don't need oligomerization.
I see. Thanks.
Okay, Zhiyong has a question, so can you unmute yourself?
Oh yeah, thanks for the great talk. Yeah, very appreciate that.
Thank you.
So I have a
Thank you.
I'm working on the human genetics, so I just googled the human variant about the STING. So in early 2014, there are New England Journal, they reported 6 patients with the heterozygous variant for the STING, and the patient showed the early onset inflammation. So it seems like it's a gain of function mutant for the inflammation, something like that. So
Yeah.
so I was wondering your C53 inhibitors could somehow to benefit the patient for that. But I just checked that the variant is not near the 53 S53 position. It's more near the 4th and the 5th transmembrane regions. So I want to hear some thoughts for that from you. Yeah, thanks.
Yeah, so that's great. And so I was want to highlight first the now people have pretty good understanding of all these STING activation mutants. A lot of them are here, around this hinge area. Like they can. So James Chen and Xue Zhang and Xiaochen Bai from the UT Southwestern saw the structure, and then they have a hypothesis that this mutant somehow mimic the conformational change induced by CGM, and that's how they induce auto activation, right? And then it's really beautiful, and so it's really the field still don't fully understand, how much the contribution come from, especially for diseases, how much the contribution come from the interferon or NF-kappa B pathway activation, how much of the pathology come from the channel related, because people just don't have the concept they have it, right? But you can imagine, I would imagine for the inflammation related, probably still the interferon or NF-kappa B induced downstream cytokine. But then, there are also reported a lot of different cell deaths, including T cell deaths or the neuronal cell death that is interferon independent. Could that actually come from the chronic activation of like ion flow? So we don't know. But I think now we have a drug. Not we have a way, at least we want to maybe specifically perturb one arm to see how much the contribution of the channel part to the downstream pathology. So I think that would be really important for the downstream. But I also want to highlight the disadvantage of our approach, because the C53 is first is a blocker of the channel, but second is also agonist for the interferon. So it's not a perfect perturbation system if you just want to block the channel part. But again, I think probably it won't be too hard for people to have a blocker just block the channel without perturbing the interferon in the future. So maybe that can be a better perturbation for the disease model research. But we are really looking forward to that, because especially a lot of neuronal cells have STING expression. And it's a mystery. People highlight a lot of the interferon function, but we wonder how much is the ion flow function there, to induce like responsible for a lot of physiology or the pathology behaviors.
Bingxu, maybe I missed this. I just hope you could also help clarify a little bit. You showed that when you apply the agonist, the STING is localized like by the super resolution imaging. It's localized at the Golgi. But before you add the agonist, are they there, or is just translocation going on? Where are they?
So STING, I didn't show, but STING normally localize a lot on mostly on ER membrane without agonist.
So there is a translocation going on induced by the agonist.
Yeah. When you get agonist,
that makes sense.
Clear. Yeah.
Also, I have a question about your screen. The screen nicely also pull out some ATPase. So I just wonder, in the beginning when you were thinking about this, why you don't hypothesize that it might be modifying the function of the ATPase that's changing the pH. Like ATPase, I know itself is regulating acidification of organelles, but why didn't you hypothesize that it is functioning through ATPase?
So first, it's even harder to prove ATPase is not involved in the processes. So first, I will not say I have like super strong evidence that it is not responsible. But indeed, people have known, from Feng Xiao's really nice work, that VATP is actually So it's a function initiator for the downstream LC3 lipidation. So basically, VATP is they sense the pH increase and then they're reassembling of the V0 and V1, and the reassembling process give you the downstream LC3 lipidation. But the reassembling process rely on the initial pH increase on this acidic organelle.
Thank you. Thank you.
Yeah. So yeah, must be something upstream there.
This is amazing. A lot of cool data and really impressive. All this imaging, all this detailed calculation you were able to do. And congratulations again. I hope you order that. Oh, okay. Sure.
Okay.
Yeah, please.
Yeah. Okay. I have another question regarding the C53, the pocket. So you think that's the proton binding pocket. So can you so I'm just wondering if you can just mutate those residues binding to proton instead of using a C53 to prove that those are the only the binding. Thanks.
Yeah, we had that data, and then we actually this was supposed the whole figure there. We removed it during the revision process. So the short answer is we can, and then we can do it, and then we did it, and then the problem is we actually identified several mutants can block more of the downstream LC3B more specifically, but the problem is these mutants, again, like C53, they not only give you the LC3 reduction, they also give you some level of autoactivation. And the problem is autoactivation itself is not a problem, but then they induce the partial sting degradation. So every time we do experiment, we have less sting to compare to.
I see.
We have some flow assay to normalize, but it's just never as clean and conclusive as C53. So we decided to remove it. But then we only did several, the saturated mutagenesis on several the sites along the channel. But if we do more, probably we can find a better mutant that only blocks the channel part without giving the autoactivation. So that's probably can answer the question more clearly. But that's really a good question. And also, just before the ending, I want to highlight that this is we're not alone, and then the field is at a stage that basically really getting there. And right after us, a very similar archive actually posted. And then also this is the email from Richard Euler that basically when I thank him for such a wonderful fundamental work, he's saying like they actually have similar hypotheses and even was in the state of testing them. So it's just like something I feel I want to share, since it's not like of course I think our team really did a really amazing work, but it's really a lot of fundamental work also bring the whole field to almost at the stage people realize that should be the case.
Thanks.
Oh, we can't hear you, Charles. We can't hear you.
Yeah.
Before we leave and before anyone has a final burning question, I just want you to have like a final comment, because I'm not familiar with the experiment data using like liposome. I just wonder how you or your collaborators think about the property as ion channel, because they might have looked at other types of canonical ion channels, like voltage gated ion channel or these other channels. Do they trigger more of a spike, or is this more leaky channel? What type of property that is, like for sting as a channel?
Simple. No, and for us, we have been thinking a lot. So first question, like related to the channel, is it only a proton channel? Can it be the calcium channel, other things? So we don't know, right? And second is like even like Shanshan mentioned, like in the beginning, is it really a voltage gated channel or is agonist gated channel? We also don't know for sure, right? So I think these are like just open questions. We just got lucky that things happen to work out in our assay. But what are actual properties as the channel itself? I think we're not expert there. We're just trying to bring as much as we can. But I think the question you mentioned will be critical for like people have known thing somehow is responsible for calcium imbalance inside the cells. Is it like a direct effect, or is maybe the voltage change somehow mess with the calcium channel? and then so we don't know.
Okay, that's amazing. I guess that's also in a few years, since you bring attention to this aspect of its function, someone will sort out, or someone in your lab or other people in the field who will maybe
I really look
solve
forward
this
to that.
mystery. Yeah. Okay, thanks again, and hope you all the best. And yeah, we will look forward to hearing from your exciting work next stage. Okay.
Oh yeah.
Thank you
All
very
right.
much for your time.
Thank you for the chance.
Bye.
Bye-bye.
Thank you.
Bye-bye.
Bye.